Hello, hello, and welcome back. Yes. Please. Please, people. Yes. If someone is still missing, shout something. Yes. Works every time. Yes, so we are in very good pace in this conference, and the next thing is the short talks. And yeah, just a reminder, the time limit is 8 minutes, sorry, 10 minutes, sorry. But yes, but I will stand up at 8, so you will see that it's the 8 minutes time point, and then you have 10 minutes wrap up and so forth. And yes, and stand here, because if you are here, no one hears you, and then there's some cameras that are recording, so your mom is also hearing, so hi mom. All right, so these are the instructions, and the last instruction is to have fun. That is the most important thing here. That's why we all are here in the conference. But enough talks from my side. I will want to introduce the first speaker, who is Vilhelm Suksi.
Thank you very much, Tuomas. All right, metabolomics. Well, we've heard much today already about single-cell transcriptomics, spatial transcriptomics. We're going to hear about orchestrating microbiome analysis and single-cell analysis and all kinds of stuff, but comparatively, maybe metabolomics support isn't as well developed in Bioconductor. And here I will first go through the background for this recent development of this notame package for Bioconductor, and then some central features, and then a sort of Bioconductor compatible workflow, as well as some future prospects. Yes. So this package was first developed in a nutritional metabolomics research group in Turku, Hanhineva Lab, and also in conjunction with this metabolomics service company, and they provide all manners of metabolomics analysis for clients. And it kind of started off at a bundle of tools. They wanted stuff to work consistently, and considering this brief overview here on the right, notame is limited to this like tabular data processing. So we have normalization and quality control, imputation, feature clustering, transformation, and scaling, as well as feature selection. So there we have univariate analysis and supervised learning tools, as well as ranking and filtering to select the most biologically relevant features. And the metabolomics field at this point uses these rather standard quality control metrics defined by Broadhurst. That's the relative standard deviation as well as the dispersion ratio. And these are based on pooled QC samples. And there are many sorts of variations of how you can pool these QC samples. But generally speaking, you want to pool these QC samples from all your biological samples. And then you can use these QC samples to correct for drift in injection order. And then there is also this very kind of well-working universal solution for normalization locally, which is called probabilistic quotient normalization. And that's included as well. And then in metabolomics, when we analyze the data, we get the feature table, but there is this redundant representation of metabolites, since in the ionization step in the instrumentation, there are some molecules are ionized into several different moieties. And we also include some data wrangling data wrangling support. So for example, simply combining batches in metabolomics, we may want the injection order to carry through the batches, also for the QC samples to make the QC-based drift correction work correctly. Yep. Yay! No! All right. Brief Adobe Acrobat commercial. Yeah. Yeah. Okay, and the Bioconductor workflow. So in the nutritional metabolomics group, which I'm associated with, they typically use MS-Dial for the sample preprocessing, and then they use notame for the tabular data preprocessing. So that's normalization and drift correction and imputation and stuff. And we use a host of quality control visualizations here. So, for example, here in the left-hand plot, the dark tile is the QC samples, and we would expect the QC samples to be identical as they are pooled from all the biological samples and sub-aliquoted. So after these drift correction and normalization procedures, we would expect the tile to appear darker, so the distance between the samples would be smaller. And then also these exploratory analysis like PCA also aid in just gaining an understanding of the structure of the data. And then for feature selection, there's a host of univariate tests which cater to most study designs. And then biological context, usually we rely on point-and-click tools, often a Metaboanalyst, and then there are databases for annotation and stuff. But now, with the Bioconductor, and especially the R4 mass spectrometry initiative, the further development of XGMS, we can use XGMS for the sample pre-processing. And then we could use notame or other Bioconductor packages which also support the same data structure, summarized experiment, for example, for batch correction and perhaps mechanism-aware imputation, where we first classify the missing values depending on their missingness mechanism. And also, of course, we may use packages from other domains where there are many methods which can be adopted for metabolomics as well. So, for example, the microbiome analysis MIA package. And then there are also some complementary metabolomics packages, such as QM tools package, which has some shared functionality, and then also unique functionality, unique strengths. And on the featured selection side, a Bioconductor has this kind of staple packages such as Limma, and then we have an extension to that also developed in Turku, and Ali is sitting there in the audience somewhere proudly, Limrots, and mixomics, for which summarized experiment support hopefully is coming. Tuomas has been working on that, so we're trying to develop the ecosystem here to cater to metabolomics using the same data structures. And then for machine learning, simple interface is microPML. And then on the biological context and annotation side, we have packages for that too, and these also are covered by the Metabonaut workflow, which will be presented in the next presentation. And some prospects. So this package is now in review in Bioconductor, and it was, as I said, a big bundle of tools. And really these functionalities are kind of separate and distinct. So we decided to split the package into notame viz and notame stats. Notame stats and notame viz, they both offer functionality which could be used in other domains as well. So we try to make it uniquely appropriate for metabolomics, or try to emphasize some metabolomics-specific functionality. So, for example, we may want to perform univariate tests for all features, also low-quality features, or at least keep them in the data structure, as that may aid in annotation and of the metabolites. We try to complement Bioconductor to make this work seamlessly and not just pile more stuff into our packages. Optimally, we want to reduce the stuff in our packages and just make the Bioconductor ecosystem work more fluently. And perhaps the chief advantage of this is easy reproducibility with the Bioconductor version management. And this would allow us to shift our focus in the package development more to metabolomics. So maybe we would focus less on notame-viz and notame-stats and more on the notame package, which provides this very metabolomics-specific functionality. And then some acknowledgements. So Broadhurst for quality metrics, and then Bioconductor, yay. And then we have notame workflow for non-targeted LCMS metabolic profiling. This is the protocol article which was published in association with this package, and it has proved useful especially as a starting point for new practitioners. And then I'd like to thank the collaborators Atte and Retu and Ville and Leo and Kati. Yes, thank you very much. Any questions?
Yes. Thank you very much. It was pretty interesting story about the hassle.
Yeah, no problem. Yeah.
Yes, time for questions.
Hi, thank you. Notame is such a cool work. You said you were going to now focus more on the Notame package by itself. Do you know what you will implement next? Or?
Well, actually, we met in Prague too for the Metabolomics conference, so there was some drift correction methodology, which was interesting there. So I think there are many developments. We always glean these developments, but then it would take more time to actually benchmark them and stuff. So we're not quite yet at that stage and deciding, filtering what to include. Yeah, probably these single functions which do one thing clearly well. Yeah, that would be the focus.
Time for a quick question.
Thank you for your nice talk. You mentioned about the integration of univariate analysis. Is the only unique statistical analysis that you implemented, you are going to implement in the package? And the second question I was having is that this package will be only for non-targeted liquid chromatography mass spectrometry, right? Is there any plan for some brand targeted metabolomics that you think about, for example, Metabonaut or Biocrates, that can be integrated, for example?
Yeah, so of course many of these methods are universally applicable and could be applied in completely other domains. Who knows, maybe the concepts are similar and stuff. And we have had some people work for targeted metabolomics datasets with the same package. And then I'd return to your first question. So, the functionality for univariate statistics, which is useful in metabolomics specifically. So that's we flag our features based on quality, but then we may want to still include them in the univariate tests, because that allows us to see which redundant metabolites might be originating from the same object, and we might choose, okay, that's the one which represents the actual metabolite we're interested in. Yeah, something like that. I hope I answered your first question too.
All right, let's thank Vilhelm once again. And the next speaker, we will continue with metabolomics. The next speaker is Philippine Louail. these
Yeah.
and these, but don't touch these.
Is there any way I can have the notes on it? No. It's
No,
fine.
I guess.
Cool. There. Wait. Nope. Yes, thank you. Hi, so I'm Philippine Louail, and we're going to continue on the coolest omics, which is metabolomics. And I will present to you a side project of my PhD, which was developing this Metabonaut website, where we kind of try to direct people into the analysis of untargeted metabolomics. So before going into the website, I want to more explain why it was created in the context in which it was created. So I'll just go back a bit into the basics, and we'll start with mass spectrometry. So mass spectrometry is a really cool technique, which essentially allows us to measure small molecules in a system, and these molecules can be peptides, lipids, metabolites, many others. So it allows us to have some high throughput possibilities, and with it, we can do, for example, targeted or untargeted metabolomics. In our lab, we focus a bit more on untargeted, which means that we will have a relative abundance of our metabolites from the sample, and it's mostly hypothesis generating and not hypothesis driven. So essentially, we have a sample and we try to figure out all of the metabolites that are in there. The main problem of mass spectrometry is that we are, by using just this technique, we are unable to distinguish between metabolites that have a similar mass to charge ratio. So in the quick example I have here is this glucose, fructose, mannose. They have the same molecular formula. They will ionize in a very similar way, which means we cannot, in the same sample, we cannot determine their relative abundance. Kind of easy to fix this. Yes. Essentially, we need to first separate the molecules before injecting them into the mass spectrometer. Here we focus on liquid chromatography, which allows us to add additional dimension, which is retention time. And now our glucose, fructose, and mannose are properly separated into 3 different peaks. Oh, sorry. Okay. So all of this to explain that LCMS is essentially a multidimensional type of data where we have to analyze the data both in the retention time dimension and the mass to charge ratio dimension. So it's a complex type of data. On top of this, we have many different setups, both in the mass spectrometry side with different type of analyzer, Orbitrap, time of flights, but also in the LC side with many, many different type of column, which have different gradient, buffers, way of separating, essentially. So all of this results in very complex type of data. On top of this, everything is more high throughput, so we have even bigger data. So this is to bring you to the point that we need tools and diverse tools in order to analyze this data properly. And in our Bioconductor, the first step into analyzing LCMS data was with the EuroBioC software, which essentially is a software to pre-process raw LCMS data. Since then, the ecosystem is now really big under the umbrella of the alpha mass spectrometry ecosystem. And we have now a bunch of tools which target different type of the analysis workflow. I don't want to go too much into it. I just want to show you some example. But for example, Spectra was created in order to properly handle the mass spectrometry data and be able to be flexible and scalable in order to deal with this complexity. And on top of this, we have some other packages that were here to expand its functionalities. And this is this allows to have different representation of the data, and again, being able to handle much larger data sets. Just to explain to you that the complexity of the data resulted in the need for more diverse tools. And now we have many of them, which target different type of the analysis, like compounding, annotation. The integration with Bioconductor allows us to have many, many different type of downstream analysis possible. And a latest focus of the alpha mass spec has been interpretability, either through implementing the capabilities of Python library or other software directly in R, or focusing on format exchanges, essentially. But one downside of this was we now have complexity of the softwares. And even though we tried to really have proper tutorials, proper documentation for everything that we develop, the main feedback that we got from the user was that it was complicated. People didn't know how to put everything together. And so the first thing that we thought about was to put everything together and have this end-to-end workflow from the raw data to the annotation, explaining everything in detail. And yeah, so we had that. But we also quickly realized, no, that was too fast. Well, we quickly realized that it was not going to be enough, essentially, because we have different contexts, design study, and we realized that we needed to be able to go more in depth into certain part of the analysis. Yeah, and some things should be done differently depending on your study design, etc. So that's why Metabonaut was born. And this end-to-end workflow didn't stay alone very long. We now have multiple vignettes that are implemented describing very different part of the analysis. And they are all written here. No. Okay. Yeah, that was a gamble, I knew it. But essentially, I will show you more images afterward. But now the current set of Metabonauts is, we have, wow, okay. Yeah, so we have now multiple vignettes that allows us to, where we describe all of these things. All these vignettes are written in Quarto. The one important thing is that we are using the same data sets to analyze to do all of these vignettes, which really gives this like coherency and consistency to the users when they go for the vignette. They know exactly which data set we're talking about, and we're trying to promote that as much as possible. Okay, so Metabonaut really aims to be educational. We try to go into thorough explanation of certain part of the data analysis, but especially in the context of untargeted metabolomics. And we try to always have some journal peer review to promote it as much as possible. And this is necessary because unfortunately in the metabolomic field, we still are lacking a bit of standardization of proper quality control, quality analysis rules. This is something that has been the focus of the field in the last few years, but there's still people that disagree on certain part of it. So it's important that when we do certain type of normalization, we're able to justify why we are doing it and also infer properly the results of everything. So it might seem redundant to people that are doing genomics or transcriptomics, but this is very important in the context of metabolomics. So another key reason why Metabonaut aims to be educational is that this dataset that we use is a real dataset, so like a subset of something that we created in the lab. So it's very much imperfect, especially a subset of it that we chose randomly. But that allows us to discuss real problems that can arise with the users and that they should look for, and things that they should look for before doing any type of analysis, because that would prevent them from losing too much time, essentially. And the last thing that makes Metabonaut kind of educational is the fact that we support a Docker setup, which is kind of perfect for a classroom environment. So each vignette is then already run, so as soon as and everything is contained in the image, so very nice for a classroom. Lastly, so I said that our mass spectrometry has been focusing lately on interpretability, and this is also reflected in Metabonaut. So one of the vignettes that we're using, we show how to use SpectriPy, which allow annotation of dataset using a Python library. And something else that we're working on is integrating the normalization and quality control aspect of notame, so the presentation you just had before. So the goal of Metabonaut is very much to have this collaborative website where everyone with different expertise can submit their vignettes and just improving the field and the way we analyze the data all together. And so there's many people to thank. The people written here are the main people that worked on the vignette in Metabonaut, and obviously all of the developers of the Bioconductor tools we are using, and the QR code linked to Metabonaut and some contact links. And yeah, if you have anything you want to contribute, any question, feedback, or whatever, don't hesitate to contact us. And obviously thanking the funding also at the bottom. Thank you for listening.
Thank you very much. Pretty damn awesome. I really like great documentation, and I have checked the documentation. It's pretty nice.
Thank you.
Discussion. Any questions? Well, maybe I can then start. Well, I'm not expert in metabolics. I'm pretty stupid in that field. But have you focused on the interpretation? Because I know that there's a lot of things that you have to a little bit.
Tweak,
yeah. So.
Yeah, very much. I mean, so we mostly show univariate analysis in the workflow for now. But again, it needs to be interpreted properly. Sometimes just like adjusting the p-value is a discussion that I've had in the field, for example. So yeah, all of this, we try to explain it as in the basics, why would you adjust and what happens if you don't adjust, for example. We also explain interpretation of everything, so like the PCAs, what happens if you do normalization that doesn't work properly, and etc. And that's something we want to expand on also. But for this, we also need more lab people because there's some question we don't realize they're asking and things like that. But yeah, we question everything and we're very critical of any results essentially. Okay,
that's nice. Time for a quick question if there is. Yes.
Thank you again for your nice talk. This package seemed to be useful. I was asking to the people who are working in biomarker discovery, where they deal with a huge number of data, because untargeted you have number of hundreds of metabolites that you have to analyze, and maybe if you are comparing disease and control, you need some kind of method of dimensional reduction, like PCA. Is that already included? Is something that you're planning to do in the future?
Yeah, I mean, yeah, the data that we work on is really large. In this workflow specifically, no, because we need to be able to run it to do the test that it's working. So this method only has 12, but we do discuss it. We do discuss that reduction and everything. Yeah, reduction of dimensionality. So yeah, this is something we discuss. And I mean, also when we run univariate analysis on thousands of features, that is also you also need to be able to explain the results properly and everything. So yeah, I don't know if that answers the question.
All right, let's thank Philippine.
Thank you very much.
All right, the next speaker is Lieven Clement.
Thank you. Okay, so I'm happy to introduce omicsGMF, which is a multi-tool for dimensionality reduction, batch correction, and imputation. And it does that for single-cell omics data, and it does that in one go. It does that simultaneously. The work that I will present is mainly the work of Alex, who is a PhD student in my group, and it was a collaboration with the Davide Risso group, with collaborators Christian, Christoph, and Leonard. And I will present their work here today. What is omicsGMF all about? It is basically a bioconductor wrapper for a package, an R package that is called scGMF, and that is a kind of a general matrix factorization method for all members of the exponential family. And that has the advantage that it can model both RNA-seq data by using negative binomials or Poisson distributions, as well as mass spectrometry-based proteomics, which is approximately Gaussian upon log2 transformation. Today, I will mainly focus on the mass spectrometry proteomics applications. So how this scGMF works is working. So we basically have a data matrix Y, and here in the columns we have the features, and in the rows we have cells or samples. And we will model this data matrix with a mean that is linked to a linear predictor via the link function of the exponential family. And we will model that mean by using cell-specific covariates, feature-specific covariates, and a bunch of latent factors. And then we basically get cell-specific scores that we can use for a low-dimensional reduction visualization of the data. And here we basically see that our cells are nicely separated according to their treatment. And the nice thing of this dimension reduction is that with this model, we can automatically correct for known covariates and batch effects, and then have a kind of a dimension reduction that is corrected for that within one go without having to optimize different steps. One of the cool features of this algorithm is that it is a very fast implementation that is using a stochastic gradient descent algorithm. And in mass spectrometry data, we often also have missing data. And the nice thing of ScGMF is that it can also impute the missing values by using the current estimates of the mean model in the current iteration. And that is doing something like EM-like algorithms. And once that we have these missing values that we can deal with, we can also exploit them for other reasons. We can also, for instance, mask certain values in the matrix and try to reconstruct them with the model, and we can then use cross-validation in order to select a number of latent factors. And this is a very cool and new feature that is not possible with competing methods. So how is omicsGMF now performing on proteomics data? So the first data set that I will show here is the Petrosius data set, and it's a single-cell proteomics data set on 525 single cells. That's a lot in mass spec, because in mass spec we have to run every cell on a single MS if we do label-free. For this data, we cannot measure proteins right away. We typically cut them into pieces, into peptides, and we were able to identify 4,435 peptides for this data set. And we see that the missingness is huge. In this data matrix, missingness is up to 60%. Competing methods will first do a kind of an imputation step and then conventional PCA for dimensional reduction. And here I will show methods in a state-of-the-art package proteomics imputation models for mass spectrometry. And if you compare that with our omicsGMF approach, we see that the cells that were treated with or without an inhibitor are much better separated in omicsGMF than in the competing methods that use a two-step approach. We will now ramp it up to a slightly more complex design. And this design is the Leduc data set. Again, single cell data. Now they have 1,500 cells from two cell lines, melanoma cells and monocytes. And the researchers here used TMT multiplexing to multiplex up to 14 cells in a single MS run. And that will, of course, cause a strong run effect, because cells that are in the same MS run will experience the same technical variability. On top of that, because they had to run hundreds of MS runs, they also had to change this LC, or the liquid chromatography column, in between, and that causes an additional large batch effect. Again, about 4,000 and so peptides were identified, and 60% of the data was missing. And the nice thing with omicsGMF is that we can include or estimate our latent factors while correcting for batch effects. And here we took the TMT run as a kind of a cell-specific covariate. And if we do so, we see that the composition nicely clusters the data according to their biology, according to the melanoma or monocyte cell type. And another thing that we also have here as a feature is that we can spot a novel cluster of interesting melanoma cells, which also have been reported in the literature. And if you use the conventional two-step approach with imputation and dimension reduction, we lose that feature, and we clearly see that we still need some additional batch correction, and that this cluster is still hidden in the data. So we mainly see a large effect of the LC batch, and then we have some effect of the cell type, and this green cluster is still not visible. This would involve additional batch correction steps, and then we need like a complex procedure, and we have to think about what do we have to do first, imputation, batch correction, or vice versa, or these steps optimized, and so on, if you put them in a pipeline. And with omicsGMF, we don't have to open this kind of worms, because we do everything simultaneously. The last data set that I will show here is a data set on bulk proteomics, so we can also work on bulk data. It's a spike-in data set, so we know the ground truth here. And we have a yeast background that is the same in all the samples. And 48 different proteins were spiked in at 5 different concentrations. And then these samples were sent in triplicate to 3 different labs, so we have 45 samples in total. And because we have bulk data, the resolution is much higher, so we now were able to identify up to 10,000 peptides, and we still have a vast amount of missingness because we have so many runs. And if you look to omicsGMF, we clearly see that omicsGMF in its first dimension is separating the samples according to the spike-in condition, and in the second dimension we see a split according to the lab. And this is also visible for our competitors, but if the competitors have a harder time in separating the low concentrations from each other. And that, of course, also has an effect on the downstream differential analysis. So if you look to the downstream differential analysis in proteomics data, we typically start from peptides. We will summarize them into protein expression values, and then we will do a differential analysis, and that's what we do here. And the plots that we have here are showing the sensitivity or the true positive rate in function of the false discovery proportion. And the state-of-the-art methods that we currently use, just use the data as they are, summarize peptides into proteins while correcting for peptide effects. And we see that they behave quite well, but they are vastly outperformed in terms of sensitivity if you use this omicsGMF imputation step before. And if you do the imputation with machine learning methods that are currently state-of-the-art in the literature, we see that they seem to perform much worse than even not imputing the data. Another thing that is also useful is to look at the log2 fold changes, because we have ground truth data. We know that the human spike-in proteins are differentially abundant. We also know their fold change. And we see that the fold change for a method that uses the data as is spot on. And we see that all the imputation approaches are inducing a bias, but the bias of omicsGMF is much lower than the bias that we see in other methods. Why is that the case? Here I will just show that. We have log2 transformed and normalized intensities for the different concentrations, and we clearly see that if the concentration increases, that the peptide intensity for the spike-in peptides is also increasing. And omicsGMF also has the same trend in the imputed values, but they are albeit a little bit lower because they are missing by low abundance. And this trend is not picked up by our competitors, and that's why they do not perform as well as what we do. Do I still have time for one slide? Yes. Okay. So another cool thing is that we also have model selection with cross-validation, and we clearly see that we can select a number of latent covariates, and we can either correct for known covariates or not, and we see for the Petrosis data set that that's not really necessary. We can pick it up with an additional latent factor. For the Leduc data set, it is important to correct for the batch effect, because then we need much less latent covariates, and we also see that the cross-validation error is much better. And for the CPTAC study, we also have seen here that it is better not to model the data in function of the known concentration, because then we tend to have like overfitting and the latent, and this is probably because this is only present in a very low number of features. So to conclude, omicsGMF is a multi-tool for simultaneous dimension reduction, batch correction, and imputation. We can apply it both to bulk and single-cell proteomics, and as well as transcriptomics. We can select the number of known covariates as well as the number of latent factors, and it generally improves upon dimension reduction and imputation. And we have two preprints out, a preprint on omicsGMF and a preprint on ScGGMF. Here you can find the proteomics work, and here you can find the applications on single cell. And we have two R and Bioconductor packages out as well. So I'm happy to take questions.
Thank you very much. One question then.
I have a question about the plots that you make. They're all square. So your principal component plots are square, and your GNMF plots are square. Does that mean that it's like when you make UMAP, that you have the same amount of variance explained on the same axes? Or that's just a choice you made? Because in principal component, usually that's wrong. We don't do it that way. We make them proportional to
Yeah.
so that you can see that distances between two points are
So
well.
the first axis is explaining more variability than the second
Is
one.
the same amount?
No, they are explaining, I think, I should check how much, but the first component is explaining more variability than the second one.
I see,
Yeah.
because
Yeah.
it's important to know when you're trying to interpret the separations.
Sure, so in the first component we have the largest source of separation.
Okay, okay.
All right. Well, very quick.
Oh, I guess it's not quick. You mentioned that they do the downstream, that they do the differential analysis downstream.
Yeah.
Could you integrate that also into the model and basically the parameter inference on your known covariates?
This is something that we are thinking about. Currently, we cannot do that yet. We should, in principle, be able to do it. For single-cell transcriptomics, it's a bit difficult because ScGMF can currently not estimate feature-specific dispersions yet. So therefore, we have not integrated it within the framework yet. But that's definitely something that we can think about.
All right. Thank you, Lieven.
And now we continue with Geraldson. Muluh, please.
Oh, yeah, this one. This works. This. Don't touch the center one.
Which one is for the light? This one?
Okay, good morning, everyone. All right, so I think this is the time where we hear a little bit about the microbiome. We've had a lot of other stuff. And I'm not sure I have a roadmap. Vince mentioned to have a roadmap, but well, I will maybe give the motivation behind this package and then maybe review some methods that we have there and then conclude. So once again, I'm Geraldson Muluh from the University of Turku at the Department of Computing. And so today I will introduce to you miaTime, which is a scalable Bioconductor framework for microbiome analysis. It analyzes changes over time. And yep, so next. Oh, next. All right, so our motivation is quite straightforward. The thing is, longitudinal microbiome data is actually, it follows like this complex analysis and it's not well standardized. And so given that the microbiome ecosystem is very, very dynamic, it responds to changes in diet and lifestyle and so on. We don't yet have like the actual standardized methods that can enable us to be able to do this analysis. The temporal patterns are very crucial for health, but most of the times the tools that we come across are usually designed for these static snapshots of the microbiome. So the motivation behind this package was to be able to have something that can enable us to be able to follow the trajectory of the changes in the microbiome over time. And so we proposed this miaTime to Bioconductor, which has been submitted and will be published very soon. It contains this native implementation that follows the Bioconductor standards. And then it also contains some robust statistical methods. And it has a quite intuitive, easy to use interface for the user. All right, so miaTime is based on some of the most common data structures that are supported by the Bioconductor, which in this case is the tree summarized experiment. And then it is also depends on some other tools that are developed in our lab, the MIA tools, if you've heard of it. We have this ecosystem of packages that enable microbiome analysis. And so miaTime depends on that, together with the tree summarized experiment in order to facilitate this temporal analysis. And in this package, we have these various methods for analyzing like the baseline divergence, the stepwise divergence, stability assessment, biomodality detection, and short-term changes. I'm sure you must be wondering what divergence here means. So divergence in this aspect, it refers to the dissimilarities between microbiome samples between different time points. And so sometimes you can have an intervention study where you have like an early reading and then a later reading, and you want to follow the trajectory of the microbiome over this time period. And so in this case of a temporal divergence, we present two types in this miaTime package. We have the baseline divergence that seeks to find the dissimilarities between samples between the first time point and the last time point, and then we also have this stepwise divergence that wants to check the dissimilarities between consecutive time points. And so we are going to take a look at these two divergences. Yep. So the baseline divergence here, as I earlier mentioned, it checks for the dissimilarities between samples at the first time point and the last time point. So given that this intervention study was conducted over several hours, so it checks from the very first hour to the last hour, and then it supports several dissimilarity metrics, such as the breaker test and the HSN, depending on the type of data that you're dealing with. It could maybe you are taking into consideration the compositionality, or perhaps maybe you just want to deal with the relative abundance. And so if we consider, for example, an intervention study where we administer diet, maybe for this study, and then we collect samples between two time points. Let's say we have two diets, let's say oat and rice, and we want to check the dissimilarities between the samples between the first time point and the second time point. This method is quite efficient and easy to use. As you can see, we provide the get baseline divergence method there. Looking at it, it's quite intuitive that you can directly apply to your data and obtain results. So we currently support subject-wise analysis and a flexible baseline definition, which means that you can also define a reference point for the baseline in your analysis. And then moving on, we have the stepwise divergence analysis, which now measures the dissimilarities between consecutive time points, not just now the first and the last time points, but also seeks to check the changes between the various consecutive time points. So you might want to check maybe what are the differences between, let's say, the beginning of the experiment and, let's say, 10 hours after, or maybe between 10 and 20 hours after, and so on. And so this is quite relevant in very high-frequency sampling data to check, because sometimes, sometimes the changes might occur somewhere within the experiment. It might not be at the start, it might not be at the end, but it might be somewhere within the, and you want to find out where exactly is this change occurring or how it is responding to the perturbation that is being administered. And then now we have these other metrics that we also calculate stability and bimodality. So the concept of stability here is not stability per se, but it's like an intermediate stability measure that relies on the correlation between various changes within the microbiome in order to get certain metrics that can tell us exactly if the microbiome is stable over the course of the experiment. Together with stability, we also have this bimodality coefficient that we are able to calculate based on the distribution of the data. It is a central moment method that enables us to get this coefficient, and it can tell us if, for example, these features are existing in two states. Sometimes we have features that can be abundant at certain times of the experiment and then become extremely rare in other times of the experiment. So we want to check and rule out this particular kind of features in the experiment, or it's like somewhat heuristic methods that enable you to do the trial and error and then point you in the right direction. So most of these methods are, what? Oh shit. Okay, yeah, these methods are clinically relevant in various studies, such as in the ecosystem health, in disease states, and for therapeutic targets. Now, a good, wow. Where was I? Sorry about it. Okay, so a good thing about this package is that it is quite interoperable and fits into the Bioconductor ecosystem of packages. And you use the standardized workflows that are compatible in Bioconductor. It also adheres to the FAIR principles of findability, accessibility, interpretability, and reusability. And it also has an active community development around it. We do have the GitHub repository where we have a lot of new ideas that are coming in, planned features. So we are also looking forward to time to event modeling, as well as users are also proposing other features that they would like to see in this particular package. And then we also have like a community on Zulip. Bioconductor moved from Slack, and it's now on Zulip. And if you type miaverse on Zulip, you will find our channel there, where if you try using the package or you run into some problems, you can often like suggest what you need or your issues there. And then we also have like numerous training materials. I think Helena was mentioning that most biologists usually want to find like the simplest questions to their problems. They want the simplest methods in order to find solutions to their problems. And so this is exactly what miaTime with its neighboring packages does. So we have this OMA book that will be presented in one of the workshops tomorrow, orchestrating microbiome analysis. I think that will be presented by Thomas tomorrow at the workshop. And he has a really nice surprise for those who will be attending. So if you want a surprise, then you should definitely check it out. And so we have these training materials that assist the users. I think, was it also Vince that mentioned that these biobooks that we provide, they are not just a package, they are not just a book, but it is a production and selection set. So it has like this consistent workflow that can enable you, it guides you, the user, to be able to perform these various analyses. And so to conclude, I am inviting you to try miaTime for your microbiome temporal analysis. And thank you.
Thank you very much, Muluh. Yes, question.
Hi, thank you. Very, very nice presentation. So it's more of a naive biological question. Is what you talked about is dynamics of microbiome as a whole, mostly. Would you say that you could identify variations or dynamics in individual species within this microbiome and relate it to the donor's health or pathology?
Yeah, I think that's exactly what it does. Like, you can be able to find these variations within the microbiome across these different time points and be able to correlate them. MIA also has some packages that enable you to like do this cross-validation between metadata of the...
And biologically, it makes some sense. I mean, there's...
Well, I am not a biologist, so I can't say exactly, but for what I can say, it's that the methods that miaTime provides are simply descriptive and more of exploratory kind of methods. They are not like very definitive, so they can like help point you in the right direction of what you're looking for.
Thank you.
Hello, thank you for the nice talk. Just one quick question about the longitudinal part. Can it work on 2 or 3 time points, like minimum, or has to be like a lot of time points?
I mean, as long as there are 2 or more time points, it should definitely work. I'm sorry, are you asking like about the computational capacity, if you can take more data?
So there are other microbiome similar tools, and they need like at least 5 or 6 time points to say it's longitudinal.
Oh.
That's why I ask.
Well, I am not sure exactly.
Right. Let's thank Muluh once more.
All right. The next talk is from Johannes Rainer.
This one and this and that, and please don't touch this.
This one? Okay. Yes. So that's the... Okay, cool. Great. So, hello everybody. Does my presentation start? Or I can also tell a joke, but maybe it's not a good idea. I don't have to push anything on this, right? So just as a first introduction, so we go back from microbiome now to mass spectrometry again, so just that you are prepared, and also a little bit away from, let's say, biology more to infrastructure work that we did in the last couple of months, maybe, or we don't see anything. I see Robert sitting there. No? Hi. Yes. Okay, great. So yes, so that's the topic I want to talk about. So it's SpectriPy, our new bioconductor package, and before actually, explaining what it all is. Okay. Am I sure I'm not allowed to put anything? Where is the mouse? No. Okay, apparently we have some issues. I thought it's a clever idea to have a PDF presentation, but apparently not really. Okay, now Robert is there trying to help out. Any other questions related to T-shirts or? Damn it. This other language. I did that for my master thesis, actually. Should I connect my computer? Or?
Windows laptop and Windows froze.
Oh, okay.
So, but no problem, we're going to...
Not a big presentation.
Yeah. Yeah. Let me think. Where do I have this? Is there pictures? No. This is somewhere else. Where's attendance? Short talks. Oh no. Where's it? Short talks. Wednesday. Where are you? This one, okay. Where is this?
I thought I do not put the PowerPoint presentation because I let them read the slides sometimes it does not work. Yeah. Yay.
But you have to change the... you have to use this one. Okay? Left and right.
Wait, but it's not showing?
Oh. It wasn't there?
No.
Wait a minute. Wait. Close. So let me just try. So this is now working. Okay, now you just have to put the source here on the left turn. Okay, let me see. It's not. No. Volviamo subire aquí, no?
Sì. Ora. Perché posso che vuole definire che? O perché in principio?
Una pantalla negra y...
He says he doesn't get the signal, but before he got it, so I don't understand what happened. Once he stopped getting this. It has an external display. This is the video. So it was working before. Yeah, I did the opening something like this. Let me just check. Okay. Okay. Great. So, so we can get started now. Federico. So, as I said, I'm going to present this package and just giving some very brief background information. So, in the last couple of years, what we have done is, as part of the R4MassSpec initiative, we have developed what I would say a quite comprehensive set of bioconductor packages that cover different aspects in the analysis of proteomics and metabolomics data analysis. They are, of course, nicely integrated with bioconductors. We are reusing core data structures, so it's what makes it then easy, what we have seen from Philly's presentation, that you can build these nice customizable workflows, combining the different functionalities from the packages, combined with the bioconductor packages, and create workflows that are really specific and targeted and tailored towards your data set and your type of analysis. So this is great. At the core, we've already heard about that. We have the Spectra package that provides the infrastructure to handle this type of data, so mass spectrometry data. And just to explain a little bit some implementation nodes on the Spectra object. So what we did is essentially completely separate the code that the user will generally use to analyze the data, which is essentially provided for the Spectra object, from all the code that's required to handle the data, to import the data, to what we call then represent the data, which is taken care of by a virtual class, which is called MS Backend. And what this essentially allows us is that we can have different ways to represent the data. So we can have the most obvious implementation would be this one, so MS Backend memory, all the data is in memory. It's a very fast way to access the data. However, in this case, also for large-scale data, it's not really possible to use it because you will not have enough memory on your computer. So for that, we have then alternative representations where we keep only part of the data in memory and retrieve the full, let's say, mass spec data, the big chunks of data from either the original data files, like in the MZR backend, or with the MS Backend SQL, we can really analyze huge data sets because all the data comes from SQL databases and we don't have the data in memory. And I'm just telling this because, in essence, for a Spectra package, the Spectra object doesn't really care where the data is stored and how the data is stored, as long as the backend can provide the data in a format that it understands it. So that's great, and we are now pretty happy, I would say, with our ecosystem, and we can analyze the data. So we say, wow, yeah, that's great, but of course there's this other thing there. You might have heard from Python, maybe, maybe not. But of course, there were also clever people in Python that did a lot of developments, and one of the main packages in Python to handle mass spectrometry data is MatchMS. It also provides the infrastructure, and on top of that, there have been some very nice, at least in my eyes, algorithms implemented like Spec2Vec, MS2 Deep Score, that are very helpful in the annotation of untargeted metabolomics data, because that's also a little bit the bottleneck at the moment to analyze the data. So then, of course, I, coming from R, was like, oh, I would like to use this methodology. How could I do that? Yes, we re-implement because we like to code, right? But that's, of course, not a good idea. So the idea was really, we don't want to re-implement what they did, but we want to reuse it. And another package that you have probably heard already is Reticulate. So that's, let's say, bridging R and Python. So it's a POSIT package that essentially translates basic R data structures to basic Python data structures and vice versa. So just the basic numeric and whatever data types there are. So what we did now is we built on top of these Reticulate and built the SpectriPy package that essentially translates mass spectrometry data structures. So we can now translate the spectra objects we have to the MatchMS and the Spectrum Utils library representations of mass spectrometry data. So that's using the same approach. So we are essentially copying the data back and forth between R and Python, which has the disadvantage that sometimes you can have two copies of the same data. So we run again into memory problems eventually. But that's why I told you a little bit about this backend thing before. What we also implemented is a MS backend Pi that essentially we keep all the data in Python, we don't have any data in R, and we just make use of these backend principles to load only the data and translate the data we need at a time from Python to R. Right, so that's, and the goal of this SpectriPy package was also that we did not want to wrap the Python code into R function and make it easy for R users to use it and to apply it, but we really wanted to allow this combined analysis that you call these algorithms, these routines in their native environment. And just some quick examples how you can do this now. So let's assume we have here this nice R code. We load Spectra and SpectriPy package. You have a Python session. You import MatchMS. And in this case, this call here loads data, mass spectrometry data from a file in MGF format. So that's a specific file format for mass spectrometry data into this mass spectrometry data representation of the MatchMS library. And we have a MGF Pi variable in the Python environment. Now, if I want to get to use and to get access to this data, in R, what I do is I create a Spectra object, say I want to use a MS backend Pi, and I just give it the name of the variable in the Python environment that keeps the data. But at this time, I'm not copying any data. So I'm just creating this, let's say, virtual representation of the data which is in Python. And in the next call here, I'm just subsetting this spectra object, or this data, which is, I think, just 100 spectra, but a subset to the first one, and want to plot it. And in essence, what the backend then does, it just translates and moves this small part of the data from Python to R, and we can have this nice visualization of a mass spectrum, like we have already seen before. And of course, we can continue the analysis, like in this case in Python. We do what's called normalization, which I think is a wrong way, but it's just scaling intensities that the maximum intensity in a spectrum is 1. So that's a typical operation that you do with fragment spectra. So we run this in Python, and since we don't have a copy of the data in R, the next time I call now my plot spectra on this same data, I get now the intensity scaled to 1. So I'm immediately seeing any changes I do to the data in Python in R without having to have additional copies. And of course, we can also turn the whole thing around, so we can load data in R. In this case, I'm getting reference library, so it's a set of reference fragment spectra that you can use for annotation. That's from MassBank. We put it into annotation hub, so we can make use of this nice resource in Bioconductor that you can get these libraries in a very easy way to just call this single line here. We do some processing, and this MB variable is now a spectra object, and that's great. So we have the data in R. If I want to use this data now in Python, what I do here is just changing the backend of my spectra object from whatever backend it's currently using. In this case, it's a SQL backend. I change to MS backend Pi, and what happens then is that this call will essentially take all the mass spectrometry data from R, moves it to Python, and then just refers to it by the variable name. And I can then continue the analysis here in Python, for example, in this case, calculating similarities of whatever data you have in Python with this reference data you just loaded in R. And of course, you can have these two sessions separately, but we have Quarto, right? So why not putting everything together in a single document? So you have then the R code chunks together with the Python code chunks, and you just do your data analysis like you did with R Markdown, just with Quarto, and you have R and Python, so the benefits from both worlds in the same file. Right, and I think that was all I wanted to say, except of course I have to thank all the people that were involved here. It's quite a bunch of contributors, also because we had in February a hackathon at the EuroBioC meeting, a developer meeting, and during that meeting, luckily there were some Python mass spectrometry people, and we refactored the whole package, and that's why it has now this backend idea and this way of analyzing the data. It's also now submitted and also accepted in Bioconductor. And here I have to thank Andres and Vince. Thanks. Because we needed to fix and sort out some issues and problems on the Bioconductor build system to get our automatic installation of the Python libraries and dependencies going, and also to have the Quarto vignette properly rendering. Yes, so that was all. Thank you. If you have any questions.
Thank you. Johannes, there's a question.
Thanks a lot for your great presentation. I have this question that there are lots of resources such as memory and stuff that you told that go back and forth from Python to R and like this. And I want to say that do you have any shared resources among these two things?
I'm not sure if it could be that there might be some data structures that Reticulate can have this shared data. So shared memory, but I'm not sure. The ones that I looked at, or at least at the time when we started developing it, there was always full copy of the data from Python to R and vice versa. And especially for the Python or for the mass spectrometry data structures, they are a little bit peculiar. They're quite tricky structures that you have to take care of. And so, yeah, maybe in future, because I've seen quite some developments in Reticulate lately, maybe there are some improvements, but at the moment, we did not see that.
Okay, so in case that if you want to do data analysis, both, for example, in R and Python for different purposes, you need to have a copy of this data.
No, with the SpectriPy, not.
Oh,
you just have it in Python.
Okay.
If you want to do the Python analysis, but then if you, let's say you're done with Python, you can move on and move all the data to R if you want, change the backend again to something that doesn't keep anything in memory. It's just a matter of some code lines that you have to do to achieve that.
Time for very quick.
Thanks for the talk. I was just curious. Here you implemented this method to keep only one copy for mass spec data, but is it generalizable if you want to create any basically alternative object for Python classes in R?
This one is really specific for this type of data, and also honestly, it's quite challenging because obviously the people in Python call the variable, so the metadata and all this way in a different way that we do. They have different support of types of metadata, so it was the biggest challenge to really match the different data structures that they are, that we are not losing information. So I would not say SpectriPy is not for generalizable use, let's say.
Thank you.
Right. Johannes, you were hiring, I guess.
Yeah, sorry, yes. So yes, I'm hiring. So if 3-year postdoc position, integration of these things into Galaxy also. So just in case.
Thanks, Johannes. And the next speaker is Florian Auer.
Okay, good morning, everyone. I want to introduce you my package RCX2. Basically, again, it's like adapting data structures to R, so this time with biological networks. And good. Just a quick reminder what biological networks are. So basically they consist of nodes, sometimes called vertices, and between them there are edges, and of course you can add additional data, usually called attributes or properties. For example, from experiments you add the data to the nodes or references to external databases, and of course you can do that for the edges as well. And on top of that, you also can do that for the network itself, for example, the title of the network or a description, or again, link to the source you got the network from. So, okay, it takes a while. So, of course, you want to do some informative stuff with the network, so basically visualize it. And either you do it individually, like telling each node what to do, but that's basically not the best idea. So there's this way of just using the data you integrated into the network as basis for your visualization. So you can do like discrete mappings. No, is it working? Yeah, discrete mappings where you specify the names, for example, and it's treated in one way, like the form of the nodes, or continuous mappings, so the size of the nodes reflects maybe the significance value. Or you do some path-through, for example, the node names are just path-through from the node name attribute. So, okay, you now have a nice, beautiful network, which looks awesome, but yeah, most of the times networks are big and difficult to explore. So you want to share it with people, so maybe you're working together with physicians, they want to have a look at it, they want to zoom in. Well, if you export it as simply as a PNG or something, it doesn't work quite well. So there are options, for example, this Index platform, which is basically Google Drive or Dropbox for biological networks. What you can do, I have this package IndexR as well. You can query the database, search some networks there, retrieve it in R, work with it, do some analysis, do some data integration. You have a beautiful network again. Use IndexR to put it back on the Index platform. So if you have it on the Index platform, it's not the end. The platform itself is a little bit more than that. You can share your networks with either certain people or even groups of people, create your own groups. But also a nice feature is you can share your networks along with your publications. So you can have a DOI, to share along in your publication, and you can reference your network there, which is good for reproducibility, because that's always an issue to find the network node again. Okay, so far I talked a lot about how to store or visualize the networks. Seriously? For some reason it doesn't work, or there's a delay. Okay, so I didn't talk about the network structure itself. So first of all, there was some years ago the Cytoscape Exchange data model, which is used by the Index platform and Cytoscape. Cytoscape is a software tool for visualizing biological networks. So it is a JSON-based format, and the main idea is to have encapsulated aspects of the network. For example, you have the nodes with some properties. You have where are the edges? The edges, which basically connect some nodes. You have the layout, like the position of the nodes in there is another aspect. And you have like the visualization as another aspect within the network. So you share the visualization along with the network. Of course, there's some metadata for managing it, but the main problem is that's data format for transmission of the network. So not basically keeping it in memory efficiently, and not for dealing with it in R. So I made this RCX package, which basically was adopting the CX format. And as you can see, there are some core aspects, which basically are the most important ones. They had to be supported, otherwise it's not a network. And then some specific ones for the visualization, especially in Cytoscape. And, well, wow. Okay, no, that's not the end. Okay, here we go. Interesting, it jumps. So this package has a lot of functionality, like the conversion from and to the JSON-based format. You can create, modify the network and its aspects. You can validate all the contents, because CX requires a specific format and implicit dependencies. And that's not really things you want to think about when you deal with it in R. You just want to have your network and work with it. Also, if you're working in R, you have some standard packages like iGraph or a Bioconductor graph package. So yeah, you want to connect to tools that use DAT packages. Of course, you can convert the network in DAT or from that. So you have a bidirectional lossless conversion. And of course, you want to visualize the network. You can do it in R as well and export the visualization even as an HTML page file. Also, the RCX package is used within NXR, of course. I left out that part, like NXR is using RCX for dealing with all the data. So yeah, that's RCX. I was talking about RCX2 initially, or at least the title of my presentation says. So let's see if that... Yeah, so basically, it has the same functionality. Yeah, nothing new actually. But in the background, there's a new development on the ECX format, which basically had breaking changes. Now it's more smooth, more dense, so the networks are not that big, so you really can send on the internet quite quickly. And there are some really breaking changes in it. For example, the nodes have now the position included as attributes, standard. The aspects, like the node attributes, edge attributes, all these attributes, well, they were kind of loosely defined. So each element within the attribute had to define its data type, its basically its data type, and that for every entry again. So now you have a declaration of all that attributes. You make a list. This attribute name is a string. This is a number, and so on. So you have consistency, and you have default values, so you don't have to encode things 100 times with a network, which really reduces the size and makes it more performant. What else? You can fragment aspects. For example, you can have nodes a couple of times, because if you do analysis, generate new nodes or new edges, you can pack them in different aspects. So before it was in one aspect, it was monolithically. Now you can transfer it one by one as it comes out of tool before, and you can use it. So also the visualization changed. Before there was a lot of Cytoscape specific elements in there. They are out. And now there's a new kind of portable Cytoscape style implemented, which works not only on Cytoscape, but also Cytoscape JS and all similar tools. So this, of course, got me thinking, like, okay, should I just simply adopt that? No, I thought like one critical point in RCX1 was that, wow, that it wasn't really feeling like using a network in R. So I kind of changed how you deal with the network, and now I introduced more like a standard way of accessing the data. Like, first of all, you create simply by a data frame. You also can do that for edge, on an edge based as well. Here it's only for nodes. Like, you see the nodes, you can access all the attributes. Along with that, you get basically a data frame out of that with all the nodes and the attributes to it. You also can use it like, using standard R stuff. You can change also the column names and the mappings. Like I said, some have data types. You can change it easily. And the mapping is done in the background. You don't have to care all the JSON stuff happening. So I mentioned that you can do also your own aspects. If you have to encode additional information for a different experiment, which has a specific format which is relevant to you, you can encode it in an own aspect, and you can basically set the conversion from and to JSON in a more easier way, so the conversion is part of the network and doesn't have to be loaded somewhere else. Also, the big point is legacy. So you have networks in the RCX format. You of course want to continue using it. So you can convert your RCX networks into RCX2 and continue using them. Yeah. So basically, there's a lot more around that. So I didn't submit it yet because I still have to fix some bugs with it. But soon, hopefully, end of this month, I can submit it to Bioconductor, and this is the existing package already in NXR, so you can use them to deal with the networks. And there's also like further stuff on the index and Cytoscape consortium, and I also wrote some useful documentation about how to use all that stuff. Yes. With that, I want to finish my talk. Open for questions. Thank you.
Thank you, Florian. Unfortunately, there is no time for questions, but you can ask in the break.
Oh, I also have a poster tomorrow, so you can also stop by there.
All right, thank you. And the last speaker is Lucas Beerland.
Right, so instructions this and that, yes, and then you can get the light from here, but don't touch the sender.
Okay, thank you. Hello everyone. I won't take up too much of your time so we can have lunch. But yeah, I've made this entire presentation, so please stay with me for a few minutes. I've been working on a package named GAMDIT, which stands for Generalized Additive Models for Differential Distributions. To illustrate my method, I'll show you an example. This is an example from a single-cell proteomics dataset, and the example is a protein called phosphodiesterase 11A, which breaks down the phosphor bonds in cells and that way regulates cyclic AMP molecules. So when I perform a conventional you told me not to touch this, but I touched it. Yes, okay. When I perform a conventional differential expression analysis on this, I get a p-value that's not significant. In this case, I used MSQROP, and MSQROP only looks at shifts in mean. And that's because MSQROP, like most other methods for differential expression, are derived from bulk proteomics. And in bulk, it makes sense to only look at shift in mean, because you don't have a lot of samples. But in these single-cell approaches, you do have a lot of samples, so you do have information to look beyond the mean, to look at entire distributions. Okay? These are the distributions you can get out of single-cell data. So that's where GAMDIT comes in. It's a method that can detect any kind of difference in distribution, not just shifts in mean. Okay? And how does it do that? It does that using Lindsay's method. And Lindsay's method is a very clever trick to recast the problem of estimating distributions, of density estimation, into a problem of Poisson regression. Okay? And how does it do that? Basically, Lindsay's method makes a histogram. It discretizes the sample space. So you have some bins. For every bin, you have an associated midpoint and a count. Counts are Poisson distributed, so you can model the counts in function of the midpoints using a Poisson generalized additive model, a GAM, using smoothers. That's why the shapes are so flexible. The model are the lines. Okay, so the model allows us to, for every midpoint, predict a count for, in this case, melanoma cells and monocytes. Okay, so, and also notice that the midpoints are the same, in this case, across both sample types. So every sample type has the same bins. So when we look at the model, we see this part. This means that there is a smoother for every sample type. So the distribution can be freely modeled. That's why it's so flexible. And we also have a offset, which is just a normalization factor. It just allows the distribution to be shifted up or down. It doesn't influence the shape. Okay? So now we know how to recast the problem of density estimation into a Poisson regression problem. We know what the model looks like. Now the question is, how can we use this model to perform inference, to compare the two distributions, to compare these smoothers? Okay? To look at that, I'll first explain what a smoother looks like. At the top part, you have the smoother for the melanoma cells. At the bottom part, you have the smoother for the monocytes. And as you can see, a smoother is basically just a linear combination of basis functions, these 5 functions. Okay? And as I explained before, the mids of both sample types are equal in this case. We've constructed it that way, which means all basis functions are the same for both sample types, which means if you want to look at the null hypothesis being, is the smoother for the monocytes equal to the smoother of the melanoma cells? In other words, are both distributions the same? This is equivalent to looking at, are all smooth parameters the same? Okay? And this is very handy because we can test this simultaneously using a wall test, a general wall test, again using the GAM framework. When we specify the right contrast matrix L and a parameter vector beta, we can perform a wall test. This is the general expression of a wall test. And this generates a p-value. This p-value reflects the difference in distribution between sample types, in this case. Okay? So when I do this for this example, I get a p-value that's very significant. Okay? But this is just a p-value. What we would like to do is we want to add some more interpretation to it. Okay? To do that, we'll go back to the histograms and the smoothers. What we will do is we will make a difference smoother. So how do we do that? We make a fine grid, and for every point in the grid, we subtract the prediction for the melanoma smoother from the prediction from the monocyte smoother, and that way we can construct a difference smoother. Looks something like that. Along with the difference smoother, as you can see, the dotted lines, we construct simultaneous confidence bands using the GAM framework. And we can use those confidence bands to look at regions, to look at differential regions, regions that drive the significant p-value. Those are the regions where the confidence band does not include 0, here indicated with the gray shades. So this is the kind of output you can generate along with a p-value. It contains a lot of information. You have the histograms, you have the model fit, and you have the differential regions that drive the significant change. Okay, so it's not just a p-value. And then finally, let's take a look at the performance of our method. We've simulated some different distributional differences, and we'll compare 3 methods. First of all, you have MSQROP, which is the conventional differential expression workflow, so only looks at shifts in mean. You have our method, GAMDIT, and you have distinct, which is the main competitor. It's also designed to detect any shift in distribution. Okay? And when we simulate just a shift in mean, we see that our method doesn't lose a lot of power as compared to a conventional differential expression workflow, which is very good, certainly when you look at some other simulations we did. We did 4 different scenarios of differences in distribution, and in the first 2, you can see that our method clearly performs better than distinct. Okay, MSQROP is not even on those 2 figures here, on those 2 curves, because there's no shift in mean in the first 2 cases. So MSQROP doesn't detect anything, which is what you want. And in the last 2 examples, you see that all methods are very comparable, which means in every scenario, our method performs comparable or better, which is nice. And then some take-home messages. When you want to look beyond mean shift and compare entire distributions, you should take a look at GAMDIT. It's not out yet, but hopefully soon. It doesn't only give you p-values, it gives you an interpretable visualization that contains a lot of information and is directly linked to the inference. It's fast and scalable. It can do 10,000 features in a few minutes, which is faster than, for example, distinct or other competitors. And it's very flexible. You can also use it to, for example, compare more than 2 groups, which for now no other differential distribution method can do. Thank you for your attention.
Thank you, Lucas.
Thank you, Lucas. Very clever. I guess this can be extended to other types of data. And if so, how big would you recommend to be the sample size? So how many samples would you advise to use?
Yeah, it definitely can be other data sets, because you basically make a histogram. And you should have enough samples to make sure you have a nice distribution.
Nice distribution, yeah.
That's
Thank
it,
you.
basically.
Thank you.
There is a question there.
Yes, thanks so much. It's very cool. If I understood correctly, you're treating each cell as an independent replicate. Is there some way to also incorporate the information that some cells might be similar to each other because they come from the same sample? So if you have like a 3 against 3 type of mouse type of thing versus 1,000 against 1,000 cell thing?
Not yet, but we're thinking about it. But not yet. We've not incorporated that for the moment.
But is there a good way to incorporate this into this framework? In an elegant way?
An elegant way? I hope so. Not yet, but we're thinking about incorporating that.
There is still one.
Hello. Great talk. I enjoyed it. I was wondering, have you ever considered about like correlation structure between the fitted coefficients in the GAM model, and does that affect, like if there is, then does that affect the results of the hypothesis testing?
Oh, that's a good question, actually. I don't think in this case, because we use thin plate regression splines, I don't think there's correlation between the model parameters. I think so. But it's a good question. I should think about it.
All right. Thank you, Lucas.
Thank you.
And while we are already clapping, let's thank all the presenters. So now we have lunch, and after the lunch we start at 13:45. So please be back.